This lesson is part 2 of an introduction to cellular respiration. In this video, I'm going to focus on aerobic respiration, specifically what happens after glycolysis with the oxidation of pyruvate, the Krebs cycle, and the electron transport chain. A reminder that the entire point of cellular respiration is to make ATP. It's to make adenosine triphosphate to power the reactions in the cell. We're going to go through a lot of complex chemistry, but this is our end goal. In the last video, I mentioned that there are two types of respiration, anaerobic and aerobic. We reviewed that anaerobic respiration is respiration without oxygen. It occurs in the cytosol and is two steps, glycolysis and lactic acid fermentation. For prokaryotes, that's the end of the story. But for eukaryotic cells, cells that use oxygen primarily for energy, they'll begin with glycolysis, skip fermentation, and instead begin the further steps of aerobic respiration. Aerobic respiration requires oxygen. It occurs in the cytosol for glycolysis, but if oxygen is present, we have further steps. Specifically, we'll be able to do pyruvate, Krebs, and the electron transport chain. Why do all of this? Because it harvests so much more ATP than glycolysis alone. A review of glycolysis. We started with one glucose molecule and ended with two pyruvate. What happened was glycolysis was initially split into 2G3P with the addition of two phosphates. Those two phosphates from ATP destabilized the molecule. We were then able to harvest out 4 ATP and 2 NADH, giving us a net yield of 2 ATP, 2 NADH, and 2 pyruvate. So we're now left with this. Our glucose has now resulted in 2 pyruvates. So what can we do with that? Well, we can do two more steps with the carbon found in the pyruvate, the oxidation of pyruvate and the Krebs cycle. Notice that carbon has not been lost yet. Our 6-carbon glucose has just been reformed into 2 3-carbon molecules. So glycolysis is only the start. Now we can do more oxidation. Specifically, we can rip those carbons off of the pyruvates, and if we have oxygen, use that to harvest even more energy. For what I'm going to cover from here on out, I just want to mention that the remaining steps occur inside the mitochondria, so it only occurs in eukaryotes. It does not occur in prokaryotes. So since we're going to be talking about the mitochondria, I want to review the structure of the mitochondria. It has a double membrane, a smooth outer membrane, and a highly folded inner membrane. That inner membrane is referred to as the cristae. Between that outer membrane and the cristae is a space we call the inner membrane space. It is filled with fluid, and the fluid inside the cristae is referred to as the matrix. There I'll find DNA and ribosomes that are unique to the mitochondria. They are separate from what is found in the nucleus. So let's look at that first step. What's going to happen to those pyruvates when they enter the mitochondria? I'm going to show you a video animation of it, and then we'll look at it in more detail.
When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation.
A reminder that oxidation means to lose electrons, a reduction is to gain. So when you see this, think what was oxidized and what was reduced.
Each pyruvate molecule is converted into a compound called Acetyl COA. In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide.
So as you saw, pyruvate is split. When it is split apart, it makes two 2-carbon compounds called acetyl CoA. Those are oxidized. That carbon is lost as CO2 gas, and then an NAD plus is reduced, gaining an electron to become NADH. Let's talk about that in more detail. So when that pyruvate enters the mitochondrial matrix, there's going to be 3 steps. The first is that carbon is lost as CO2. We're then able to reduce an NAD plus and make it into NADH, so we're banking some electron carriers, which we can use later to harvest for energy, and we're producing 2 acetyl CoA. To show you what those molecules actually look like, here you can see the 3-carbon pyruvate on the left when it enters into the mitochondria, it's going to attach to an enzyme called coenzyme A, and that'll cause the formation of acetyl CoA. In that process, we reduce an NAD+ to make NADH, and we oxidize a carbon off in the form of CO2. Every time you breathe out, portion of that CO2 is the CO2 that you are breathing out as a direct result of this oxidation of pyruvate. So cool, now we have acetyl CoA. There's still more carbon we can harvest to get even more energy than a little bit of NADH with a process called the Krebs cycle. The Krebs cycle is named such because it was discovered by Hans Krebs, but there is a movement to no longer name processes after scientists, so oftentimes it's also referred to as being the citric acid cycle. Why? We'll see in a moment. This happens in the mitochondrial matrix, and it's going to break down that acetyl CoA into electron carriers and ATP. There's a lot of steps in this process, so I want you to keep in mind the goal of this step is to extract electron carriers and energy from acetyl CoA. That is the whole point of everything you're about to see, is just to bank electron carriers and energy from acetyl CoA. So to look at it from far away, the Krebs cycle is a cycle. It's going to end the same way it begins. We have two carbon acetyl CoAs. They're going to attach to a 4-carbon compound called oxyl acetate, and they're going to form a 6-carbon compound called citrate. This is citric acid. That's why it's often referred to as being the citric acid cycle. As we go through the cycle, you're going to see that carbons are removed in the form of CO2. That CO2 is breathed out. So all the carbon now that we brought in, those 6 carbons from glucose, are now gone. We lost 2 in the oxidation of pyruvate, and now we're losing 4, because this happens twice, because there's 2 acetyl CoAs, with the Krebs cycle. And then we'll regenerate what happened in the beginning to return to the cycle. Now there's more steps than this to it, so I'm going to play that video, and then we'll talk about it in greater detail.
The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water.
So notice it's showing you those carbons that are lost that I mentioned before, but it's also generating a lot of electron carriers and a little bit of ATP.
The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule.
That's a huge gain. We're getting a ton of NADH and FADH. We're putting money in the bank that we can use later to make ATP.
In order to understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage, the electron transport chain.
And we'll do that after looking at the Krebs cycle again. So the Krebs cycle, we have the 2-acetyl CoA going in. They're going to form citrate. As that CO2 is lost, we're actually going to be able to bank a little NADH. Again, when that CO2 is lost, we're going to get a little more NADH, and we're going to make some ATP along the way, and another electron carrier called FADH. And as we further regenerate to return to oxyl acetate at the beginning of the cycle, we again make more NADH. So with this one spin, I got 3 NADH, 1 FADH, and 1 ATP. Keep in mind, this happens twice. So we're actually losing 4 CO2, we're getting 6 NADH, 2 FADH, and 2 ATP. That is a huge gain compared to the other steps that we saw before. So what's happened? We have fully oxidized glucose. We started with 6 carbons and we've lost them all as CO2. But we've only ended up with 4 ATP so far. That's not enough. What was the whole point of all this? It's to put money in the bank. If we are able to get enough electron carriers, we're going to be able to use those later on to generate ATP. To give you a preview of how that is, This enzyme is called ATP synthase. ATP synthase is what's going to generate most of the ATP in cellular respiration. How does it do that? Well, if you can create a concentration gradient of hydrogen ions, put a lot of hydrogen here and very little there, those hydrogen ions can diffuse through ATP synthase, and that energy is used to generate ATP. Now, where would I find a bunch of hydrogens laying around to build a concentration gradient? Right here with my electron carriers, the H on NAD and the H's on FAD. The whole point of this is to get those hydrogens to power ATP synthase to make ATP. Here again, you can see ATP synthase. The whole goal is to build that concentration gradient. But we haven't done that yet. We need to go to the last step. We have no carbon remaining, but we got a bunch of electron carriers that we can use at something called the electron transport chain. Accounting for ATP so far, we got 2 in glycolysis, 2 in the Krebs cycle. That's only 4 ATP. We need a lot more than that. For perspective, one working muscle working for 1 second requires 10 million ATP. That's a lot. We're going to have to do a lot more than just glycolysis in the Krebs cycle to make that happen. There's got to be a better way. And there is. The electron transport chain is going to be able to utilize all those electron carriers and make up to 36 ATP. We only made 4 so far, so 36 is a huge gain. Also notice we haven't talked about oxygen at all yet. I thought you needed oxygen for aerobic respiration. You do in this last and final step. So this is going to happen in the mitochondria, so I want to familiarize you with the anatomy of where the electron transport chain is going to do its thing. So here you're seeing the inner membrane space, the cristae, and the matrix. So there's the inner membrane space, and here's the mitochondrial matrix. What you see is a traditional plasma membrane. You can see the phospholipids here, and you can see proteins that are integral with them. These proteins have specific functions. This is NADH dehydrogenase. It dehydranases NADH. This is cytochrome BC complex, and this is cytochrome C oxidase complex. So now we can finally put those electron carriers to work. Something to keep in mind before I show you the animation of this step. All of those hydrogens we've been getting with the electron carriers, that is just 1 proton and 1 electron. During the electron transport chain, the cell is actually going to be able to split that apart. It's going to be able to split the electron from the hydrogen. So let's see it animated, and then we'll look at it in detail.
Is a series of membrane-bound carriers in the mitochondria that pass electrons from one to another. As the electrons are transferred between the membrane proteins, the cell is able to capture energy and use it to produce ATP molecules. Proteins in the chain pump hydrogen ions across a membrane. When the hydrogen ions flow back across the membrane through an ATP synthase complex, ATP is synthesized by the enzyme ATP synthase. Oxygen acts as the terminal electron acceptor. By accepting electrons, oxygen is reduced to form water, a byproduct of the electron transport chain. All the high-energy electron carriers from the previous stages of cellular respiration bring their electrons into the chain. From this, the bulk of ATP from the entirety of cellular respiration is produced, a net of 32 to 36 ATP.
So that was a lot to take in. Let's unpack it again. So we're going to be able now to use those electron carriers. What will happen is, let's look first at the case of NADH. NADH will go up to NADH dehydrogenase and the hydrogen will be removed. When that happens, there's 2 different events. The first is an electron is removed from the hydrogen. That electron powers the pumping of the hydrogen ion, the H+, up into the inner membrane space. Remember, our goal is to make a concentration gradient so we can shove it through ATP synthase and make ATP. That electron is going to continue down the chain. That's why we call this the electron transport chain. When it does that, it continues to power further pumping of hydrogens up. Why? Again, concentration gradient. That's our goal. Now, these electrons aren't going to move on their own. FADH is going to do the same process. What's moving those electrons down the chain is an attraction to oxygen. This is why you're breathing in oxygen. We haven't gotten there yet, but we finally got there. Oxygen is going to attract electrons down the chain. And as it gobbles up those electrons, it's going to also bond with free-floating hydrogens to form water. This is the water that's formed as a waste product because of cellular respiration. So we're just building that proton gradient. We're breaking open our piggy banks. The electrons move down the chain to oxygen, which is called the final electron acceptor, because it's accepting all of those electrons, and it's being used to pump hydrogens up into the intermembrane space. By doing that, we now have our concentration gradient and can have those hydrogens diffuse through ATP synthase and make ATP. Finally, we did it. That proton gradient is going to go through ATP synthase and going to make up to 36 ATPs. That's huge. That gets us the energy we need to move things like muscles and have complex organisms like yourself. Just as a point of vocabulary, I want to mention to you that when chemicals diffuse when ions diffuse across a membrane, sometimes it's referred to as chemiosmosis, or the osmosis of chemicals. The diffusion of hydrogen ions through ATP synthase is a case of chemiosmosis. So oftentimes you'll hear people say electron transport chain or chemiosmosis interchangeably. Just so you know, electron transport chain is this entire process. Chemiosmosis is a specific term for the movement of ions across a membrane. So let's review that all again. We started with glycolysis and got 2-pyruvate. And some ATP. Those 2-pyruvate are oxidized, making a little bit of NADH that we can use later, and we're left with 2-acetyl COA. CO2 goes out as gas. That acetyl COA is broken down the Krebs cycle to make a ton more electron acceptors. The remaining carbon is lost as CO2, and we make a little bit of ATP. Once that arrives at the electron transport chain, we harvest those electrons. They're attracted down the chain towards oxygen as our final electron acceptor. The oxygen uses those electrons to make water. The movement of those electrons powers the pumping of hydrogens up into the intermembrane space. Why? Well, now I have a concentration gradient. There's more hydrogens out than there are in. So through chemiosmosis, those hydrogen ions will diffuse into the intermembrane space, and that will power the turbine of the enzyme to generate ATP, upwards to 36. Oxygen was that final electron acceptor, so keep in mind, what happens if oxygen isn't available? That's a bad thing for you. Obviously, you need it to survive. But why? Well, if that oxygen isn't there for the chain, things will get backed up. Those electrons won't be able to move down the chain. Our electron carriers won't be able to unload their hydrogens. We get no proton gradient, so we have no ATP. The cell runs out of energy and you die. This is what happens in cases of cyanide poisoning. Cyanide attaches where oxygen normally does, preventing the movement of electrons, no proton gradient, no ATP. So we did it. We made 2 ATP in glycolysis, we made 2 in the Krebs cycle, and 36 in the electron transport chain for an average total of 40. Now the number does vary based on the cell. So in summary of cellular respiration, glucose and oxygen in, CO2 and water out, and we made 40 ATP. Glucose is oxidized in the CO2, it's losing electrons. Remember, it was oxidized in the oxidation of pyruvate and further broken down in the Krebs cycle, whereas oxygen is reduced. That oxygen picks up hydrogens and forms water. If you can answer these questions, then you're in a good place for your understanding of cellular respiration. Can you explain where the glucose came from for a heterotroph or an autotroph? Can you explain where the oxygen came from? Where the CO2 came from? There's 2 places for that. Where did the CO2 go? Where did the water come from? Where did the ATP come from? And what else is produced that isn't listed in this equation? I hope this was helpful in your understanding of cellular respiration, and I'll see you next time.